Good morning. Welcome to the Precision Medicine Summit. It's my pleasure to welcome you to Columbia campus. Before we get started, I do want to recognize a few individuals. We have with us today Curator John Sunvold, thank you. Curator David Steelman and John Phillips will be joining us shortly. We also have two chancellors with us, Chancellor Alexander Cartwright, as well as Chancellor Molly Agrawal, and today's his first day as Chancellor UMKC, so welcome. I also want to recognize the organizers, including Mark McIntosh, Elizabeth Laboa, Sheila Grant, Chris Piers, and Jenny Parker from Mizzou, Lawrence Dreyfus from UMKC, as well as Chris Spilling from UMSL. This is truly a system-wide activity because it is a high priority for all of us in the UM system. The theme for today's summit is personalized medicine, with an emphasis on cardiovascular, neurological, and cancer research. The overarching plan or theme for PM is to really take the fundamental research from your laboratories and then translate that to effective treatments as well as devices. But instead of having a serial approach, our goal is to gather together the basic scientists, clinicians, and entrepreneurs so that we can have an accelerated path to take that technology to the marketplace. Recently, back in November of 2017, the Board of Curators of the UM system identified the translational precision medicine complex as the highest capital priority for the UM system. But the TPMC initiative is more than just about building a building. It's really a call to action for all of us at the UM system, the state of Missouri, and the nation to address pressing health issues. That work really begins today. And this summit will illustrate the breadth, depth, and the quality of our faculty members from all 4 campuses. And together, we're going to seek your input to make investments that we are going to make in both equipment, facilities, as well as seed grants. Mizzou, under the direction of Chancellor Cartwright, will lead this initiative, but we expect strong partnerships from the outstanding faculty members to ensure that we have meaningful collaborations that result in extramural funding and additional prestige to the university, but more importantly, making a difference by creating treatments and devices that help with the pressing health issues. So I want to thank all of you for joining us. I think this summit will lead to some wonderful opportunities for collaboration, and I want to thank you for being part of this journey with us today. Thank you. Now, it's my great opportunity to introduce Dr. Mark McIntosh, who helped to organize this summit, which is a second in a series of summits. Mark?
Thank you, Dr. Choi. And let me add my welcome to all of you for being here this morning. This is quite an impressive turnout. Let me start by suggesting that what you're going to see today, I want to be very brief because what you're really here to see is this very exciting menu of speakers who will provide us with a short but very impactful breadth of research topics related to precision medicine or personalized medicine. Before I do that, I want to thank Elizabeth Laboa and Jenny Parker in particular. Jenny, by the way, deserves a huge round of applause. She set this whole thing up. I also want to thank Chris Piers and Sheila Grant, who have been working with me very closely on several projects, including projects like this, which bring all of our campuses together to talk about research areas that will drive our future going forward. I want to thank all 4 universities for sharing your insights and your innovative faculty with us this morning. And as Dr. Choi pointed out, the objective of this is for those faculty, really, to get to know each other, because it's from those kinds of collisions that we create new opportunities for collaboration. So our goal is to use the TPMC initiative to create these collaborations across the state, leading to new discoveries and new applications of basic science toward effective precision medicine outcomes. We will begin with a vision of what the TPMC can be and what it can mean not only to the University of Missouri, but to the state and the nation in terms of translating research into application. Elizabeth Laboa will help us envision what is possible with the creation of this critical investment in research infrastructure. She will take you through a video journey of what the TPMC could be and enlist your participation in this important university objective. Then, the good part, you'll be exposed to the research that will drive this vision toward reality, as 15 faculty from across the 4 universities will give you a brief taste of what is possible. We will use this opportunity to launch further discussion and collaboration across the UM system, building interdisciplinary research teams to meet the challenges of translating our research efforts into actionable solutions and possible treatments for human and animal diseases. This is just the beginning. There are many more such opportunities to come. So now it is my pleasure to get this program moving forward by introducing Dean Elizabeth Laboa from the MU College of Engineering.
Thank you so much, Dr. McIntosh and President Choi, and thank you so much, everyone, for coming. I'm so excited to see all of you here in the audience with interest in this. This is going to be an incredible initiative. And at the end, I've just got a few slides. I do want to help you all envision what this can really be. And then at the end, I have a couple of videos to show you. But truly, the Translational Precision Medicine Complex is going to put our system and the incredible faculty, our scientists, both clinical and purely academic and researchers together in one place to address precision medicine and diseases and ways to treat those diseases in cancer, neuro, and vascular in a way that we truly believe cannot be done anywhere else. So why translational precision medicine? One, I know that is a mouthful of a name, so let's break it down a little bit. Translational medicine, as many of you who work in this field know that this has been a priority of the NIH. It is a way that we bring researchers and clinicians together in a multidisciplinary collaborative setting, really supported by the most not just advanced tools that we think about from the standpoint of the research we do in wet labs and in biolabs, but also truly taking into account the fact that the human genome was mapped in 2003. And since then, we have also begun to just really start to understand the influence and impact of epigenetics. So how we might have cancer and how it might be treated on an individual basis. It can be a function of our genes. It can also, though, as we're starting to understand even more, in particular with the incredible strengths in the omics across our system, genomics, proteomics, metabolomics, that it also is intently a function of the epigenetics, our environment and our lifestyle. And so what we want to do in the Translational Precision Medicine Complex is actually truly take the research that's occurring and translate that benchtop to bedside. And as I mentioned, this is a focus area of the NIH where no longer do we want to just be producing papers. We need to really translate the work that we're doing to help patients and to advance human health. So for the Translational Precision Medicine Complex, I'm going to show you an image in the next slide of how this facility will be laid out distinctly differently than what you have probably seen in typical research facilities. But it's really a way to depart from traditional department-based research and approaches and truly be interdisciplinary and collaborative. Through this initiative, we do not expect that we will just be a leader in the state. We expect we will be a leader in the nation, a leader in the globe in precision medicine. We want to both enhance and improve our recruitment and retention, but also we will be able to attract the most sought-after researchers, the world-class faculty around the world. And as you will see at the working lunch today, which at the end of this presentation, I'll give you a little more detail on the actual logistics of how today will go. But we are truly going to maximize the external grant funding opportunities that we can and should be doing, not just at Mizzou, but across our system. I mentioned the next generation DNA sequencing and an emphasis on the omics. Because of our incredible strengths, not just in clinical research and wet lab research and bioresearch, but also in informatics, we can advance our utilization of big data analytics for precision medicine applications that we truly believe cannot be matched elsewhere. And we're going to focus on very targeted therapies, one for cancer, but we also are going to look at the vascular opportunities. President Choi mentioned cardiovascular, but we also are looking at vascular related to regenerative medicine and tissue engineering. We will also, of course, as I said, this is an initiative and has not been mentioned yet, but I'll show it in an upcoming slide. This is an initiative not just with MU and but also with the VA healthcare system. I don't know that David Isaacs, the director of the VA hospital, was able to make it today, but he is incredibly interested in working hand in hand with us with the completion of this facility and also in collaborative research to drive the therapeutics that come out of it. So I am up here speaking, and I have the honor to speak and represent MU, but this is obviously not just one college or one university. I really want to emphasize that all of the planning that has been happening on this campus to date is a collaboration between engineering, the School of Medicine. We have Dean Patrick de la Fontaine here representing the School of Medicine. He has some incredible faculty presenting today. The School of Veterinary Medicine, Dean Carolyn Henry is here and has also been a part of all the planning. The College of Agriculture, Food, and Natural Resources, Dean Chris Daubert is here. And I don't see Pat Oker, Dean of Arts and Science, but Chris Pierce, our Associate Dean of Research, is here. And also the Veterans Administration. And behind the work that happens here, when I show you what this facility will look like, you will see how we also really focus and include the aspects of economic development that will occur as a function of this facility. So here's just a very basic schematic of a lot of faculty in the audience who know what these typical research buildings look like. We're used to seeing what's on the left. We say, okay, well, maybe in the basement we would have a vivarium, which would be small animals, large animals, and then the next floor, well, we'd have some core labs that multiple individuals can use. These can be service centers. And then typically you split things up based on, okay, maybe we're going to have this computational, the biological, the chemical, and biomaterials. Very different approach here. While we continue to have the vivarium on the bottom for both small and large animals, we're also going to have the most advanced imaging where we can truly look at this also from a One Health perspective. I know Mark McIntosh has talked a lot about getting the 7 Tesla. I've even heard 11 Tesla. I don't know that Chancellor had been aware of that until I mentioned that in a meeting yesterday. He said, wait a second. But we can really have the most advanced PET, SPECT, CT imaging capabilities to really take the imaging data and use that for diagnostics and therapeutics. And you're going to hear some incredible talks about the animal models today in the TED Talks. We're going to have the state-of-the-art training grounds for graduate students and postdocs. And what I'm going to show you next, and part of what you see in those core labs, good manufacturing practices, clean room, and a cave too. And I'm going to show you an example in a video of what that can look like. I'm sure a lot of you saw in the press recently, Bimal Balakrishnan. Are you here, Bimal? He might have gone over to, oh, he's over there. Bimal Balakrishnan is the director, and he is also the soon-to-be department chair of Architectural Studies and Human Environmental Sciences College of the Immersive Visualization Lab. He is going to show at the end of the lunch today an incredible virtual reality entree into what it looks like to walk through a molecule. What I want to show you is what we want to have in the translational precision medicine complex. It's currently what's called a cave 2, a computer-assisted visualization. And this is what it would look like if you had a top view. People in there can just have the same type of glasses you put on when you want to go see a movie in 3D at the movie theater. And what you can, if Jenny will go ahead and start that. Let me show you a little video of what this could look like. But before you start it, a truly immersive environment, multiple collaborators. So if you think about someone who's a clinician working and trying to explain to me, a dumb engineer, what this molecule is that they're working on, I can walk in with the glasses and see, oh, I'm surrounded by this molecule. Oh, so this is what you mean. Oh, this is what CRISPR-Cas9 means when you talk about SNPs. So we can all come together with very different, disparate backgrounds and truly speak the same language. Jenny, will you go ahead and start?
Cave 2 is a 24 feet wide and 8 feet tall, large-scale hybrid reality environment. It combines the benefits of both scalable resolution display walls and Cave virtual reality systems to create a seamless 2D, 3D environment that supports both information-rich analysis as well as virtual reality simulation exploration at a resolution matching human visual acuity. The walls of Cave 2 are built from 72 near-seamless, passive, stereo, off-axis optimized 3D LCD panels. This provides users with a 320-degree panoramic environment for displaying information at 37 megapixels in 3D or 74 megapixels in 2D with the horizontal visual acuity of 2020, almost 10 times the 3D resolution of the original Cave that we invented in 1992.
So I'm going to stop it there. It goes on quite a bit longer, but I don't want to cut into any of our talks. But so what the Cave 2 really does is going to be a state-of-the-art facility that advances through really advanced imaging technologies, ways for us to communicate and collaborate and think out of the box on how we're coming up with these precision medicine solutions. The last video I want to show you to help really envision what the translational precision medicine complex is next. And then I'm going to go into some logistics of how this is going to run today and then turn it over to our incredible co-moderators.
In Columbia, the University of Missouri provides global leadership through a world-class education in medicine, animal science, engineering, business, law, education, and liberal arts, all from a single campus.
Now, as the world draws even closer together, it's time to find strengths in our differences and converge, to collaborate, to integrate, to innovate, and to change the world.
Bringing these aspirations to life, the Translational Precision Medicine Complex, where researchers and clinicians work together to create the next generation of biomedical solutions.
Great minds in engineering, medicine, and vet science share spaces where new ideas collide and can join.
This nucleus of knowledge pulls in research resources from around the world, and the most promising ideas advance from concept through pilot-scale manufacturing. From benchtop to bedside, all from inside the TPMC.
Okay, I've given just a little hint of what's to come, and I'm going to stop there, but first I'm going to give some logistics. I want to, if you hadn't had the opportunity to pick up the agenda outside, there's multiple left of what these are, of the full day of what this looks like. But here's what's going to happen. In the first half, Dr. Sheila Grant, who's the Associate Dean for Research of College of Engineering and faculty in bioengineering and in College of Agriculture, Food, and Natural Resources, is going to moderate the first session. The speakers have been given very specific templates, and they've been given examples of 5-minute TED-style talks that are exceptional. The moderators want to remind the speakers, which I know they have done multiple times, that they will be stopped at 5 minutes. So they have 5-minute TED-style talks to really show the greatest work, and then at the first set, once they're done, they will line up here, so we will not do Q&A until they're done speaking, and the first set of speakers will be here for 10 to 15 minutes to answer questions. That will be Dr. Sheila Grant leading that. Then Dr. Chris Pierce will come up. Oh, and they also wanted me to remind the speakers that they will have this great iPad that will be staring in your face, counting down the 5 minutes. So then Chris Pierce will come up, Associate Dean for Research in Arts and Sciences. He will moderate the next set of speakers, and then we will have a panel. After that, we're going to go to an incredible lunch, which will truly be a working collaborative lunch. All these faculty who have heard each other speak, there will be on the tables run by incredible grant writers. And the grant writer in my college who is initiated from the College of Engineering is Shelley Hilton. But Shelley, we have grant writers from multiple colleges, right? There are printouts of the major funding initiatives that are happening in precision medicine. They will sit down at the tables with the faculty and lead them through some opportunities to really come together and do some collaborative major initiatives. So that will happen at lunch. After lunch, we are going to go over to Bimal Balakrishnan's lab, the Immersive Visualization Laboratory, which he also provided a trifold handout of how he has now set up his lab. I went over and looked at it last night. It's incredible. It's set up even better than he had for the governor's visit last week. You're going to love this. You're going to walk through a molecule. You're going to be completely immersed in precision medicine. So that's what will happen after lunch. So with that, I think the speakers will be thrilled if I finish a little early and turn it over. Here you go. Yes, there you go.
Thank you, Elizabeth. So, welcome everyone. I'd like to introduce our first speaker, Dr. Jeff Bryan. He is a veterinary oncologist leading comparative cancer research in the College of Veterinary Medicine.
Thank you. Since the war on cancer was declared in the 1970s, we have made progress in survival of cancer. But I think looking at these representative curves, both for adult cancers on the left and pediatric cancers on the right, we can agree that even a 15% chance of dying from your cancer is not progress enough. In the effort to advance this and erase this deficit of death, incredibly sophisticated mouse models and in silico and in vitro models have been developed. Unfortunately, in spite of all of their sophistication, a small percentage of drugs that make it through those models ever actually make it into the clinic to treat human patients. Those of us in comparative oncology, veterinarians who are trained in oncology and see animal patients in our hospital every day, believe that these genetically outbred, naturally occurring models of cancer that happen in our patients are exactly the model that help us improve our ability to better test drugs, get more of them into the clinic, and save more lives. Our hypothesis is that the optimal models for these cancers are literally standing beside us at the other end of the leash. And until we go further with them, we're not going to go as far as we want to go. On the MU campus, we have a remarkably unique resource of having a veterinary college with a medical school that has a cancer center with a long history of research. We have a college of engineering. We have amazing chemists, and we have this jewel of a research reactor that is truly unparalleled throughout the entire world. If we want to compete, and we compete with the research reactor, we have already won because there is no one that can do what we can do. And right across the street from that stands a building that we can move researchers into to bring together chemists and biologists and trialists to help develop the drugs that will improve our imaging and our therapy. And we have already done that on this campus. In the 1980s, a group collaborated using dogs. You may not know that one of the deadliest cancers in children, which is illustrated here, this bone cancer, is actually one of the most common cancers that we see in our companion dogs, and it's equally deadly. And this group tested a radioactive drug in dogs that for this bone cancer to try to treat it and see if they could be successful. And in fact, that led to the FDA-approved product Quadramet. And all of the Quadramet used in the Western Hemisphere is produced right here in Columbia, Missouri. This history can lead us to greater innovation in the future. The campus made an incredible investment in the PET Imaging Center and purchased this Toshiba Celestion scanner on the left. This is the newest PET CT platform on the market, and it's currently housed over in the Veterinary Health Center. On this scanner, we are able to use traditional agents like fluorine 18, fluorodeoxyglucose, or just radioactive glucose, to look at tumor metabolism. This is a dog that has a lung cancer that has metastasized throughout its spine and throughout its body. And in fact, this fluorine 18 is the only isotope that is seen on every Toshiba Celestion and most clinical scanners throughout the world. However, ours already sees not only fluorine 18, but copper-64, yttrium-90, and iodine-124. And we have a collaboration with the Canon Toshiba company that we're going to have 15 isotopes visualizable on this platform before the end of the year. And this is a remarkable move forward that no one else can match. And it leads to collaborations like we had with the Stowers Institute's action arm, Biomed Valley Discoveries. They brought to us a hypoxia-seeking bacteria, one that looks for low oxygen environments, like we find in tumors, and they wanted to know how we could make this work better. This is a patient that was in that trial, and this is the patient's tumor. And what you're seeing, produced by Dr. Meitz, is an image of two different tracers overlaid in the same image. The green is the metabolic region of the tumor, and the purple is the hypoxic or low oxygen region of the tumor. We were able to show why and how we could target this low oxygen environment. We helped them understand in a complex, naturally occurring model how much more complicated tumors are than we expected. And they also wanted us to produce for them an imaging agent that imaged bacteria where they were dividing in the tumor. And this turned out to be our greatest success, and it was our greatest failure. What we identified was that not only could this agent identify bacteria within the tumor, but it was more effective at identifying mitochondria throughout muscle. And in fact, this saved them $14 million of a $15 million human project by simply investing about $1 million as part of a dog project. Finally, we're looking at ALS, or Lou Gehrig's disease. By tracking the disappearance of a receptor in the central nervous system, we can stage Lou Gehrig's patients more effectively. And what you're looking at is a dog with Lou Gehrig's, and we can translate into the humans. We can do this in plants, but this is what matters. The man and his dog, who both have prostate cancer in common, by understanding how we can better treat Touché, we can help Jim with his tumor too. And MU uniquely has the resources to make this a reality.
Okay, next up we have Dr. Tom Spencer.
All right, howdy, good morning. Good morning. I'm not going to talk about a dog. Here we go. So although dogs do reproduce. So today I'd like to share some work that we're doing in terms of at Mizzou about reproduction and health, particularly both in men and women and farm animals. So fertility is a grand challenge to both humans as well as food animals. So infertility is common in humans. About 17% of couples will experience infertility or fertility complications. Pregnancy complications are also very common in humans. About 20% of the 6 million pregnancies in the U.S. will experience life-threatening pregnancy complications. And this is impacted by the rise in obesity, diabetes, as well as maternal age. And it impacts both the long-term health and well-being of babies as well as their mother. Fertility regulation is also an essential component of reproductive health for both men and women. There's essentially been no developments in this since the advent of the birth control in the 1960s. So in essence, non-invasive diagnostics and imaging are significant gaps in our ability to identify and treat fertility problems in humans. So if you didn't know, fertility is also a grand challenge to animals as well. So pregnancy loss is very common in domestic animals. Both cattle and pigs exhibit similar rates of pregnancy loss to humans. Pregnancy rates are particularly low in dairy cattle. Only about 18 to 20% of animals get pregnant on a single cycle, and pregnancy loss is greater than 50% in pigs. So this is a complex process. It requires multiple genes as well as epigenetics, and it also impacts the long-term health and welfare of offspring and their mothers. So fertility and pregnancy loss are actually the key components that determine the profitability and sustainability of animal production agriculture enterprises, and an increased understanding of the genetic and physiological underpinnings of fertility and pregnancy loss is a significant gap in our existing knowledge. So we have significant gaps both in human as well as animals in terms of fertility and reproduction. So in terms of solutions for this problem or grand challenge, it particularly requires a One Health type of approach where you have animal scientists, veterinary scientists, reproductive scientists, and clinicians, as well as sociologists, economists, and philosophers to tackle this complex issue. We need to focus on prevention, intervention, and invention, and also on personal and societal impacts, both in humans as well as agricultural animals. So the good thing is we have the opportunity to create unique animal models of human disease through genome engineering of large and laboratory animals. I know Randy Prather will talk about that later. We are world leaders in terms of this gene editing technology here at Mizzou. We also can use this knowledge to improve animal production through selective breeding, that is using genomics on a personalized scale at the farm level, as well as genetic engineering, which is really the translational impact and solution for both human and animal health. So I'll give you a few examples of success that we've had at Mizzou as well as elsewhere. Teams at Mizzou have identified genes that impact both reproduction and production in cattle. This has led to a genotyping platform called the GGP F250 from Neogen. This can be used to customize breeding for both sire and dams to produce genetically elite animals. And obviously, we also need more precision medicine to help solve these fertility problems. And so in humans, there's an endometrial biopsy test available now where you can determine the best time to implant an embryo for assisted reproduction. And actually, kind of the holy grail would be to test, to develop a blood DNA test like we have for fetal developmental abnormalities, but be able to do this in terms of both predicting fertility as well as pregnancy complications. So we have a lot of opportunities at Mizzou. So I can see the translational precision medicine complex providing a nexus of engineering, genomics, informatics, immunology, medicine, metagenomics, neuroscience, nutrition, and physiology. All the things that impact both reproductive health for men as well as women. And this type of approach, particularly using genetically engineered tissues as well as bioengineering, would allow us to conduct research in difficult areas. It's really impossible ethically to study pregnant women in terms of biopsies during pregnancy. This just can't work. That's why you need animal models. And we're kind of fortunate that we're developing the TPMC, and we also have a National Center for Applied Reproduction and Genomics in the Animal Science Department that also is leveraged with veterinary medicine. And we have a kind of an emerging Mizzou Institute for Women's Health Research that's being championed by obstetrics and gynecology in the School of Medicine. And so we think that we can use these critical new technologies and the knowledge that we'll have at the Precision Medicine Complex to leverage these important areas of human and animal health. Thank you.
Thank you. Our next speaker is Randy Prather.
All right, good morning. Today we're going to talk about pigs. All right, so solutions to many diseases often require models, as we heard about just recently, and pigs are already a good model for many conditions in the human. This includes bioengineering, cancer, cardiovascular disease. Pigs are trained to do certain things, and you can use them to evaluate cognitive behavior. They're used for cutaneous pharmacology. Their skin is very similar to humans. They're used in diabetes. The pig responds to a pathogen in terms of genes that it turns on in response to that pathogen, very similar to humans in contrast to rodents. Pigs are used quite extensively for nutrition studies, especially infant formulas. The pig eye has the right distribution of rods and cones, not exactly what humans have, but quite similar to humans, and they're used a lot for ophthalmology studies. Pigs are used for physiology and, again, regenerative medicine. So when you decide on what kind of a model you want, there's a number of things you need to take into consideration. One is availability. There's lots of rodents around, they're cheap, but they don't always answer the question. The phenotype counts. If you take the same mutation that occurs in humans and that causes cystic fibrosis and put it in mice, it's a chloride ion transporter, and you can block the chloride ion transport, but you don't get the meconium ileus. About 15% of humans born with cystic fibrosis have a blocked intestine, a mucus plug in their intestine. They have a blocked gallbladder, they have a blocked bile duct, they have blocked pancreatic duct. In fact, humans that have cystic fibrosis, a percentage of them have to take their pancreatic enzymes orally with their meals because they can't get it from their pancreas. They'll get liver lesions, and if they get through all that, then they grow up and get lung disease. Well, if you take that same mutation that occurs in people and put it in pigs, 100% of them have all those symptoms. So now the physicians finally have something they can go in and monitor invasively development and progression of disease. The physiology counts. An example here might be producing pharmaceuticals. We have pigs that produce human coagulation factor 8 and 9 in their milk. Well, why would you do that? Because the pig mammary gland actually puts the right secondary post-translational modifications, specifically carboxylations, on those molecules so that they're bioactive. You can have a herd of about 50 or 60 animals supply the world's need of some of these pharmaceuticals to treat people that have, for example, hemophilia. Size counts. If you're developing tools and devices to treat people, pigs are pretty similar in size to humans. If you want to develop some of those in mice, well, you have to scale up. Genome counts. We now have the technology to put any genetic modification you want into pigs, and we can make any human model that you want to make. Additionally, and maybe most important, when you go to the FDA, they're starting now to say, well, I'm glad you did this in rodents. We need to move this to another large animal, maybe a dog or a pig, before we can go into the clinics. So we've done genetic engineering to make pigs even more valuable as models. So we've made pigs that have a gene in that we can turn cancer on in any cell type that you want. We have pigs that develop cardiovascular disease. We have pigs that get cystic fibrosis, diabetes. We've made Fanconi anemia pigs, muscular dystrophy pigs, phenylketonuria pigs. You say, well, why the pig? For every one of these, there's a reason for it. The phenylketonuria is kind of interesting because the mice, they don't develop any of the cognitive disorders, the neurological disorders. Hopefully the pigs will develop those. We've made those pigs here and already done some imaging with them. Prader-Willi syndrome, this is a deletion of an imprint control region, and it results in overeating in humans. The mice don't have the overeating. We're hopeful these pigs will have the overeating. Regenerative medicine, we have pigs that we've genetically altered so that they will not reject human cells when human cells are transferred to them. So now if you have some stem cells and you have the therapy that you want to test safety and efficacy, we can put those in pigs and you can monitor how the cells are delivered. Are they delivered safely? Does it actually work? Retinitis pigmentosa. This is a really good example for regenerative medicine because if you have one mutation, vitamin A is good for you. If you have another mutation in that same gene, the rhodopsin gene, vitamin A doesn't do anything for you. If you have a third mutation, a different mutation in the rhodopsin gene, and you take vitamin A, that's bad for you. We are now making pigs that have some of these very specific mutations so that we have personalized medicine, so that we can develop these animals that have your genetic mutation, are making your protein, and we can test treatments and therapies on that. We have green pigs at the University of Missouri. You might not know that. They fluoresce. They have the gene from jellyfish, and that's used for cell tracking studies. These are actually the most popular pigs that we distribute because people want them to be able to track cells. So if you're using stem cells and you want to say, well, did this stem cell actually facilitate the repair? You can use these because they're marked, and you can turn on the UV light, and they'll fluoresce. You may not know, but the University of Missouri owns the intellectual property on the base genetic modification that's needed for xenotransplantation. Pig organs into humans. We actually have pig kidneys that we've sent to pigs that we've sent to Emory, and kidneys have gone into primates, and these kidneys have been life-supporting for over 500 days. So this is very promising. And again, I already talked about the pharmaceutical production, and that is spelled right. So here at the University of Missouri, we've made over 65 different genetic modifications in pigs, mainly as part of the National Swine Resource and Research Center, the only NIH-funded swine center in the nation. So again, for some genetic diseases, human physiologies, the preferred solution is a pig. I already gave you the example of cystic fibrosis. We talked about xenotransplantation. Again, we have the National Swine Resource and Research Center here at the University of Missouri that serves as a resource for the NIH community. We do model curation. Somebody else may make a genetic modification in a pig. We get it to the University of Missouri, and we curate it, and then we distribute to other NIH researchers that need it. We do health monitoring, new health model creation. We distribute these cells. For example, we've sent cells to Europe. We've genetically engineered cells here. We had the pig. We sent the cells to Europe. They did somatic cell nuclear transfer to create their pig there. We've made pigs here that we've actually shipped to Korea. Live animal, put them on the plane, or send them up to Chicago, put them on the plane, and ship them to Korea. We provide training and workshops. We are really positioned well for this Center of Precision Medicine. I'll end with that.
So our next speaker is John Spertus.
Thank you so much. I'm actually thrilled to hear so much of the vision is about trying to actually move our discoveries into practice. And I'd like to talk with you about what we're doing at UMKC along this journey. And it's important to have some context of what healthcare and hospitals are facing today, which is a tremendous focus on the need to improve the value of healthcare. Perhaps not such a complicated equation, but it turns out it's incredibly difficult to deliver healthcare in a highly valuable way on each and every patient, each and every day, across each and every provider. And, much of precision medicine is thinking a lot about sort of what stratifies the risk of a population of patients into those who do really well in green and those who do poorly in red. And so we focus a lot on genetics and pharmacogenomics. We focus a lot on new imaging or biomarkers that might separate risk. But it's important to recognize that we also have clinical risk models that are, in essence, precision medicine, using readily available data to identify who benefits most and who doesn't. And I would put to you that this Precision Medicine Institute should not just be focusing on discovery, but it needs to be focusing on translation. How do we move the knowledge that we generate here at the University of Missouri into routine clinical care so patients actually benefit? And so I'm going to give you one example of the need for this kind of work. So there's a therapy used in angioplasty, which is opening up blocked arteries in the heart to lower bleeding. And when it's used in patients at very low risk for bleeding, there's very little value. But when it's used at patients at high risk for bleeding, it prevents bleeding, saves lives, and saves the hospital money. And so we conducted a 9-center study to implement precision medicine. And this was at the Mayo Clinic and Barnes Jewish Hospital and Baylor and a lot of premier centers around the country. And what we did is we had 140 doctors doing angioplasty. And if they were applying the knowledge that I shared with you just a moment ago, you would expect that patients at low risk for bleeding, they would not use much of a very expensive anticoagulant. And in patients at high risk for bleeding, they would use it in everyone. And when we looked at the actual practice patterns of these excellent doctors at these excellent institutions, this is the practice pattern we saw. It's far different than what you would expect because the doctors didn't know what to do. And in fact, if you think about it and your mom needed angioplasty and she had a 10% risk of bleeding, you wouldn't want one bleeding avoidance strategy. You would want at least 2 to be done to minimize her risk of bleeding and dying. And yet, when she goes and you refer her to one of the best centers, to Mayo Clinic or to Baylor or wherever, the person who does her angioplasty is a random event. And so instead of getting 1 or 2 bleeding avoidance strategies, her chance of getting any bleeding avoidance strategy ranges from 1 to 100%. This is not acceptable, and precision medicine can be the way to do it, but it requires integrating it into routine clinical care. And so we said, why not replace the consent form with one that's generated for each and every patient that embeds in it their particular risk of bleeding so the patient and the doctor can know and can manage them appropriately. This idea of moving precision medicine into routine clinical care is the great challenge for the entire field. And it makes a big difference. This is data down the road at Barnes Jewish Hospital. They implemented our tools, and their bleeding rates went from 8 to 10% to 2%. Their acute kidney injury rates went from just under 8% to just over 2%. Their rates of inappropriate angioplasty and the ability to send people home the same day skyrocketed through the use of precision medicine. And the hospital saved almost $4 million in 2014 alone. And so in Kansas City and at UMKC, we are now trying to create the entire infrastructure to test and develop and deliver new strategies for precision medicine. We are working with a partner, the Kansas City Area Life Sciences Initiative, to bring all of the hospitals in our region to collaborate together. We are then having those hospitals identify what is the most important quality improvement initiatives that you have over the next 2 to 3 years. And we are then working with researchers throughout our region as part of our component of the regional CTSA out of KU to be able to match researchers to hospitals' needs so that we can launch and evaluate new programs to redesign healthcare delivery to improve value and often integrating precision medicine. And so this idea, all the talk that happens about precision medicine, its wonderful future, to me doesn't mean much unless it's turned to action. And I'm not the first person from Missouri to say this. When sitting on the congressional floor, Congressman Van Dever is known to have said, I come from the state that raises corn and cotton and cockleburs and Democrats, and frothy eloquence, just talk, does not, neither convinces nor satisfies me. I'm from Missouri. And this is the origin of us as the Show Me State. And it's my belief that this Precision Medicine Institute needs to be focused primarily on what it's going to deliver to society if we really want to be a world leader. Thank you very much.
Very nice, John. Thank you. So our next speaker is Dr. Mark Hofmann.
Great. Thank you. So at Children's Mercy, we are leaders in the application of genomics to patient care. We have a genome center that has sequenced diagnostically, not in research mode, thousands of children's genomes to reach diagnosis for children who would not be diagnosed without the application of this technology. But that's not what I'm going to talk about. Today, I'm going to talk about what happens outside of our genome. So within the announcement of the Precision Medicine Initiative, there's a recognition that the environment, the patient's environment, is also significant. Many of us talk about it, but we're not really doing that much to consider the impact of the environment on patient care. So I'm going to share an initiative that we're doing that begins to lay the foundation for that. Typical provider encounter can be 8 to 10 minutes. Much of that is spent documenting information into the electronic health record and just doing your typical assessment of the patient. There's very little time to apply energy towards learning more about the patient's environment. So we're doing an initiative that helps the provider at their fingertips have information about the patient's socioeconomic background, potentially then paving the way for other work around environmental exposure. So the Robert Wood Johnson Foundation did a study that demonstrated this very disturbing fact that where you live strongly correlates with how long you live. Within about a 3-mile stretch of US 71, there's a 14-year difference in average life expectancy. So to me, that's just extremely disturbing, and genetics can't explain that. So we need to also be looking at other things that can address this type of issue. So I like because we don't have enough ohms, I like the term the envirome. And the exposome is another term that's used. In my view, that addresses half of the equation. What are we exposed to? But I also think social determinants, what do we have access to, is also equally important for improving healthcare. There are large-scale data sets available already that can relate to the envirome. So social determinants, food desert data, socioeconomic data. There's environmental data sources that are already out there, but the providers they don't have time or energy to go actively seeking that information during those short patient encounters. So at Children's Mercy, we've done a front-to-back demonstration project where we've taken public data sources, loaded them into a web service, beginning with the USDA food desert data and the census data. And then we geocode our patients. So we take their address and convert it into a geocoded value that can be used to query these data sources from the context of the electronic health record. And so as a provider's interacting with a patient, we ping the web service and it passes back this context information to be viewed in the setting of the electronic health record. And food desert is the example that we focused on. So if you're not familiar with food deserts, it's the concept that at the census tract level, an area that has a combination of low income and low access to high quality food sources is considered to be a food desert. And so if you're treating a patient with diabetes and you tell them go buy fresh vegetables, but they don't have a car and they don't have ready access to a grocery store, that doesn't really help them, but you don't always know that. So a graduate student in informatics at UMKC worked with me on this We did some focus groups to evaluate what information is interesting and useful to providers at the point of care. And so we did some mock-ups. We did focus groups with providers from Children's Mercy, Truman Medical Center, and showed them some examples of how we could present the food desert data information to them. The key feedback from those focus groups was we don't want all of the graphics. We just want to know yes or no. Does this child live in a food desert? So we've taken that feedback at Children's Mercy and implemented front to back this solution in our production environment. So now the providers at Children's Mercy can, at their fingertips, determine whether or not the child they're treating lives in a food desert. And this is easily scalable to really a variety of other information sources that are available in public data. We think this can transform the conversation between the provider and their patient without requiring the provider to leap out of their comfortable workflow. And I think this is a great example of how translational precision medicine based on data can be easily implemented into clinical practice. The barrier to this is so much lower than the barrier of developing a new treatment, a new diagnostic. And so I think that making existing data more available is a really significant part of translational precision medicine. So thank you.
Thank you, Mark. So our next presenter is George Goko.
Thank you very much for the opportunity to talk to you a little bit about what we're doing. We've been working for a number of years now in the area of antibiotic resistance. You probably are all aware that there's a worldwide crisis, and the World Health Organization, CDC, have both issued enormous reports describing the potential consequences. In 2013, more than 2 million people admitted to hospitals acquired infections there, nosocomial infections or HAP infections, and 23,000 of those were mortal. For a number of years, there's been a decline in the amount of antibiotics that are being produced, and this is for a lot of reasons, but in part because of FDA regulations. What we've done is to develop a new approach, trying to develop non-resistant antibiotics. We've developed a family of synthetic ionophores. Ionophores are just compounds that conduct ions. They also function as antimicrobials. We've rationally designed these compounds so that they will do a lot of the things that natural proteins will do as channel formers. And you can see a schematic of one of our compounds here where it allows ions to pass through a simple bilayer. Of course, bacterial bilayers are much more complicated than that. Our compounds are not only drugs themselves, they kill bacteria at low concentrations, but at much lower concentrations, they also augment the efficacy of FDA-approved antibiotics, and they can reverse resistance. So if we start with a resistant organism and we treat it with the antibiotic to which it's resistant, nothing happens. If we treat it with our ionophore at a very low concentration, lower than its efficacy as a drug, also nothing happens. But if we combine the low concentration, non-toxic concentration with the antibiotic, now we have a reversal of resistance. The ionophores that we've found, and when I say we, it's mostly been studies done by the NIH Preclinical Services Organization. Our compounds are most active against MRSA, multidrug-resistant Staph aureus, and vancomycin-resistant organisms. They recover antibiotic potency, and we have determined this using microbiologists and cell biologists. The way it works is simply that these compounds make the membrane more permeable so that the drugs can get in more easily. They also inhibit efflux pumps, which is the natural way that the organism pumps out those awful antibiotics. So they can get in more easily, they can't get out, and so they kill better. We did a study on a tetracycline-resistant E. coli, and we used in the study the very closely related minocycline. You can see within 15 days it was highly resistant. Our ionophores showed no resistance, and in fact, a typical course of antibiotics is 5 to 8 days. So this is a very promising result. We also have, using contract research organizations, we have had meta-analysis, bioavailability, and a bunch of other information. This is a preliminary study that a colleague of mine who was a former student, now a professor at Notre Dame, looked at the activity of the ionophore as a drug itself. And you can see it was more potent than norfloxacin used at its appropriate concentration. The thigh infection is nearly gone here, and it is slightly abated here. So we formed a company called Upaya Pharmaceuticals. Upaya is a word that means health, and we have secured this by a bunch of patent applications. We're currently collaborating with people skilled in organic synthesis, microbiology, medicinal chemists, and so on. And this is my team. Dr. Mohit Patel is our microbiologist. Dr. Saida Nagin is our director of research. She's a PhD in chemistry. My son is a Mizzou-trained engineer who has experience in business, and he's keeping us afloat. And we think that the studies that would be performed here or could be performed here would help us with collaborators, would help us find new talent, and would be a good way to secure the interactions with the university.
Thank you, George. So our next speaker is Tim Glass.
All right, thank you. I'm here to represent both Department of Chemistry at Mizzou and the MU Research Reactor. And the way we interact with this precision medicine is through medicinal and pharmaceutical chemistry. And so let me take you a little bit through what we do. Medicinal chemistry is just what we typically think about in terms of making new drugs. So this is what pharmaceutical companies do day in and day out. But in academic research, we typically don't focus on that. We don't compete with pharma, but we end up looking more at, what are new drug targets? We try to figure out what the chemical basis of disease is and see how we can get in there and change it and to understand it better. And oftentimes this can lead to the opportunity to make new drugs, but really the focus is on understanding the disease. Now, one of the famous examples in chemistry is a professor at Northwestern, Rick Silverman, many years ago was looking at the basis of chronic pain. Why do some nerves turn on and not turn off? And so he was making some compounds to try to understand that. And one of them turned out to really relieve chronic pain. And so they took this to the clinic, and it became the drug we now know as Lyrica, which is a multi-billion-dollar per year drug. And just for fun, the background of this slide shows you a picture of the building that Professor Silverman built for Northwestern University using his own proceeds from this drug. So these can have big impacts on both the university and, in our case, on the state. We do medicinal chemistry of this type in our department, but the real opportunities for Mizzou are in radiopharmaceuticals because we have the MU Research Reactor. So here are 4 FDA-approved pharmaceutical agents that the MU Reactor has helped push through the FDA approval process. We're the only university that can say that. Now, a radiopharmaceutical is generally speaking just a drug that happens to be radioactive. Some of these can be used for imaging. So, for example, there are agents that can be used to image cancer, so you can see where it is in the body. But many of them can be used for treatment. And so of the 4, this is an imaging agent. These are all treatment agents. And I'm going to use Lutathera as my sort of example. This was developed as a collaboration, a very recent collaboration between the MU Research Reactor and this company, Advanced Accelerator Applications. And what it does is it uses what we call targeted radiotherapy. And so we all know that cancer can be treated by radiation. And that is a way of causing tumors to shrink and die. But that also damages healthy tissue around the tumor. And so one thing we can do to avoid this is we can take a radioactive element and link it to a chemical group that basically sticks to a tumor cell. Then you can inject this into a patient. It will circulate in the blood until it finds the tumor, and then it will stick. And then if you pick the radioactive element properly so that it has only a short-range radiation, then you can damage only the tumor and kill the tumor and leave the healthy tissue around it healthy. So in this particular example, the radioactive element that they used was lutetium-177 that is currently produced at MU. In fact, MU is the only provider of that radioactive element. But the rest of the drug was developed by this company that was recently purchased by Novartis for $3.9 billion. We can do that here at Mizzou if we can get the people with the right expertise together under the same roof to make things like this happen. And that's the reason why we're very interested in this translational precision medicine initiative. So where are the opportunities at MU? So the research reactor has one initiative now where they're making what they call theranostic pairs. They take the same radioactive element, but they have one isotope that's an imaging element and another isotope that's a therapy element. And so now you can develop a drug that will target a cancer, a specific kind of cancer. You could then use the imaging element to see where the cancer is in the body. And here's a picture of a rat taken at MU where the tumor is lit up by one of these imaging agents. And if the doctor, if this was in a patient, if the doctor was happy that that drug was targeting only the tumor, then they could switch to the therapy agent to deliver the radiation that would kill that tumor. This is really the essence of precision medicine. And this is one of the kinds of opportunities we have at MU to really focus on. And this TPMC initiative really allows us to take the collaborations that already exist between chemistry and the research reactor and then marry them with collaborations with VetMed to help do this in animal models, as well as medicine in radiology and oncology to really take these into people. So this is the reason why we're very excited about what the opportunities this initiative can really provide. Thank you.
Thank you, Tim. So our next speaker is Gerald Wyckoff.
Thank you. So I'm excited to be here today to talk about what we're doing and put it into context to the TPMC. So I'm at UMKC in the School of Biological Sciences, and one of the things I want to talk about is data sharing, which is a dirty word among some people because they don't like sharing data. Because there's a fear that if you don't know what's in your data and somebody else figures it out before you, there's something wrong with you, right? And even though this data is sitting in places that nobody's doing anything with it, and it can continue to sit there forever, people don't want to give it up because of that fear of missing out, of that FOMO. So how do we fix that? Well, one of the things that we're working on is to work across the state line with Kansas State University to help set up a clearinghouse for this data. And this is One Health data, which you've heard about before, so I don't have to explain it and belabor it. So we're establishing the One Data project to do this, and we actually established this at the start. January of this year is when we really formally kicked this off. So this One Health clearinghouse is actually, in the center of it is a database, and that database is large, and you don't want to look at it. But it's housed around all of the people here that you see listed that we're working with. And in talking to those groups, the most important one we started working with was Rare KC, which is a rare disease advocacy group in Kansas City that looks across all rare diseases and tries to advocate for the patients and their families. And in doing that, what they explained was that it's great to talk about natural animal models of disease, but their concern was about the awareness of these diseases, because what you find out is that rare disease is a common problem. Okay, up here you see this list of diseases that you're all familiar with and worry about. There's cancer in green, okay? There's the opioid epidemic in orange. Those are the number of people affected, all right? HIVs in gray. And at that little tiny sliver at the top you can't even see is Lyme disease, which gets so much attention that you feel like you're going to go outside and get a tick bite and die, right? That's rare diseases, okay? Take them all, stack them up, there's rare disease. Rare disease, even though they're rare, are common problems because there's 8,000 of them. So even if each one is only affecting a small number of individuals, collectively they hurt us greatly as a society. Now, that's a bad enough problem. But what the director of the rare disease group told me was even worse, because this is the diagnostic time for some of those common diseases that we worry about, okay? There's HIV, if you want to think about this from a full perspective, it's 90 days. You talk about sepsis, it's down there, strep is down there. That's 1 month, okay? That's your time for 1 month. So you can look at this and say, yeah, some of those look worse than others. Okay, there's rare disease. The average time to diagnose a rare disease in the US is 4.8 years, 1,700 days. Okay? That's 1,700 days of agony for the family and those patients. And that's why we call this the diagnostic odyssey. This isn't a neutral term, right? Odysseus suffered, right, on his way back. And so do these families and their patients. So when we were thinking about all the things that we could do, solving this diagnostic odyssey is something we decided to take on. And that's why I'm talking about it here today. Because even though we're at the start of this journey, I want to bring it to the attention of the TPMC because I feel like this is where we can make an impact. Our approach is to ask the patients what happened during their diagnostic odyssey and catalog that, both by setting up a methodology for them to share their information and looking at the metadata when they do it, and querying them directly when we actually look and ask them what happened with the survey tool, and then verify that with EMR. So we go from metadata, which has a lot of data but less rigor, to EMR at the bottom, which has lots of rigor, but we get a lot less data, okay? So this is a big data approach, not just because of the amount of data, but because of the span of data that we're looking at. And we're setting up now in all three of these areas. We have students working on this. Why am I here? What do I need from you guys? Well, this is what I need. Your expertise, your partnership, your collaboration, your patience, and I need awareness of this problem. Because we're only going to solve this for the patients that suffer for this in Missouri and move this solution into the clinic by working together. So let's bring Odysseus home. Let's end the Odyssey. Okay? So thank you for your attention.
So now we're going to have a Q&A time, but I just want to remind you guys that there are bathrooms, restrooms located both at the outside the auditorium up on top as well as the bottom. So I'm going to ask our speakers to come back up here. So we have about 10, 15 minutes of time for the audience to ask questions. Oh, okay, great. So they are available to answer any of your questions.
Hi, Craig Franklin, Department of Veterinary Pathobiology at the College of Vet Med. I want to throw another omics in. Get your thoughts on how we might incorporate the metagenome into all of these types of projects. Pretty much there's so much growing evidence that the microbial communities that we live with can impact all of these diseases, and how might we incorporate those into these types of studies in the TPMC?
Who wants to take a shot at this?
I think certainly, I mean, if you look at like one of the major fertility problems in dairy cattle is endometritis, obviously the metagenome is an important aspect of that. There's a lot of really kind of new and emerging evidence that actually the fetus is not in a sterile environment. It actually has a microbiome even in the fetal brain. And so the concept that what you eat in terms of maternal health could affect actually the baby in utero because of the microbiome is kind of a really emerging area that's a little contentious, but certainly can be explored, particularly with some animal models. And obviously, you know, any type of health and welfare of a domestic ruminant all the way to a pig or non-ruminant is going to require a healthy metagenome in terms of for lifelong productivity and sustainability.
And I would just add to that of course, you know, as you well know, the the naturally occurring disease models that we have, not just cancers, but diabetes and heart disease and things like that pass through the veterinary health center every day, offer opportunities to look at that clinically. One of the concerns that I still have about metagenomics working with your group and also with researchers in our hospital working in this is that a lot of what we have right now are associations with outcome. And causation is something we need to get to eventually. And so I think that using both basic models and then correlating those with naturally occurring models is going to be really critical to know what is real world relevant.
Well, in our efforts, we're obviously concerned about how the gut in particular responds to various bacteria, antibacterials. And so the problem is we don't want to kill everything, and we would like to know what is a response of various bacteria. Can we find genetic markers that will let us know our compound will not attack certain microbes that are essential? So that's one thing we're looking for collaborators to see if we can start to get a sense of how can we figure out what the microbiome is in the gut so that we don't have the terrible side effects of the most potent drugs.
I mean, to me, I'm a clinical cardiologist. I could hardly care less about the metagenome, but the truth of the matter is that the promise of this Precision Medicine Institute is to bring multiple people together so that you can bring in something we never thought of, and we could think about ways to try and study observationally and then experimentally its potential impact on care and outcomes. And so to me, we don't right now, if you wanted to collaborate with me, you wouldn't even know I existed, or anybody else on this panel probably. And so if there was a centralized place where we were having sort of discussions about how best to advance knowledge or care, to be able to come and represent your perspective to that other diverse group of stakeholders creates the essence of what team science is all about. And so without that infrastructure, you're not going to get nearly as far in trying to advance your philosophy as if an infrastructure like the Precision Medicine Institute exists.
Hi, I'm Leslie Lyons from the College of Veterinary Medicine. I just wanted to also point out, and maybe we want to broaden our thoughts a little bit, that all of our agricultural and companion animal species also help the welfare and benefits of humans from a non-medical point of view as well. And so we often shoot ourselves in the foot from the point of view that I've spent my life trying to make domestic cats into biomedical models for human disease and seeking NIH funding in that regard. But also, let's just think about the health of our companion animals. There's not a Mayo Clinic for cats and dogs, and maybe this facility can help realize some of those potentials that we could do that here also at MU if we maybe get partnerships from of our pet food companies and groups that are interested in animal welfare and animal health in just itself.
Did anyone want to respond?
Certainly, as the veterinarian on the panel, I completely agree with you. But I think that and it's important that we consider that. I think that we sometimes miss opportunities in veterinary health and working with some of the industry that exists along the animal health corridor of Missouri, and recognizing that while sometimes the initial outcome may really look largely animal focused, it may have greater impact on human health as well, not only through disease modeling, but also simple human-animal interaction. And there's a lot of data now showing that that's important, and that brings in the Tiger Institute and other initiatives that we have that I think looking at those things are really critical. We also are seeing things like new sources of heart disease in grain-free diets that have been thought to be commercially, they were commercially produced, they thought they were complete, and now we're finding that dogs in which a particular type of heart disease never existed suddenly exists. How many of the people that are suffering from these heart disease from surprising reasons are actually on a diagnostic odyssey waiting to discover the deficiency in their diet or the environmental interaction that has led to what they have. And perhaps by looking at our animals, we see the same things with them as the sentinels for that.
John Phillips, a curator and an enthusiastic supporter of this concept for collaboration. So there are different patient populations, demographics, that are served by each of our 4 campuses. The demographics are terribly different, and there are specialties on each of the 4 campuses that could and should blend into the collaboration at the center. You know, dentistry in Kansas City, nuclear here in Columbia, and so forth. I think it was a representative from UMKC who mentioned the territoriality of holding on to your data. So to what extent does that represent an impediment to a collaboration between the campuses and the disciplines in order to make that data available, and also worrying a little bit about the HIPAA issues as well? Is that an impediment that we can overcome, or is the big data that is generated on each campus but currently unconnected, and the libraries, which are also separate, even what you subscribe to is separate? How do we get through that, or is it feasible?
And sure, I don't know if Jerry Parker's here, but we're doing some work with Jerry related to the Cerner Health Facts data. Children's Mercy, MU, Truman all contribute de-identified electronic health record to that. And we're working towards a relationship where we can have better visibility into which site is contributing which data into that without unmasking patient IDs, without creating competitive risks. And then we think that'll will help with some data sharing that's already happening, but making it a more valuable resource that to all of the participating sites.
And I'll say one thing that we do share that does make it a little bit easier is we share the same lawyers. And I hate to say that, but it's totally true. Very often when you talk to researchers, they'll say, I want to give you this data. And then you sit down and you go through an agreement, and it stops, and it can take 6 months or a year to actually sort out how to share that. And the hope is that since we are all in the same pond, we can work that out better amongst ourselves than we could when we go outside.
I mean, I would also say that, you know, we've collaborated with, you know, Todd Vogel in vascular surgery here. We've mentored a number of the cardiologists here. You know, we're working with Jerry Parker on implementing the precision medicine tools at this hospital. But the truth is, these are all ad hoc, sort of one-off connections that, you know, through Brownian motion we've connected. Having a really university system-wide conduit for being able to nurture these kinds of collaborations would make it a whole lot easier for us going forward than it's been going past. And despite sort of the challenges, barriers, and lack of organization, there are some nice success stories, but not nearly what there could be with, I think, the Precision Medicine Institute really moving forward.
I'd like to comment on your question about the libraries. I have mentioned a number of times over the years that I've been here the fact that we don't have common access to all the libraries in our system is an impediment to research generally. And I think if we could get some kind of a uniform system across all of the campuses where we all had direct access, it would be a tremendous benefit for us. And it would also be a benefit for the startup companies that are not formally associated with the university, but which came from the university, who no longer have literature access.
So very good points. We have another question up there.
Hello, I'm Rebecca Johnson. I'm a Presidential Engagement Fellow for the system, also a professor in both veterinary medicine and nursing here at MU. And I would like to thank all of you for your splendid work and your presentations. I would like to say the Animal Health Corridor is called the Animal Health Corridor, not the Animal Medicine Corridor. I would like to follow along on Dr. Lyon's comments, which include that we need to think of this more broadly. There are many interventions that are producing wonderful health benefits that are not chemical-based, that are not genomic-based, that are based in the social sciences, and I urge us not to have a purely disease-oriented medical focus on this center, and I'm eager to participate in that effort. Thank you.
Okay, thank you for a very nice Q&A session. So, we still have time? Okay. One more question then.
So the most recent definition of precision science was best illustrated by John Spertus's diagram of the green people being cured and the red people being not being cured. And I'm wondering, when we talk about mice and rats and dogs and pigs, when do we make the transition from an inbred model, or at least sampling a small number of animals, to the point where we start asking how many different genetic variations in a model system do we have to test before we're ready to go to clinical trials with a drug that may only work on 10% of the people? So I'm curious, where in the translation pipeline do we actually employ precision medicine?
Who wants to take that question?
Well, I think that there already are some examples of this, like the MATCH trial, for example, where patients' cancers are being treated based on the genetic profiles and mutations rather than, this drug is for this type of disease. And I think that's important. There's been some work by the Comparative Oncology Trials Consortium of the NIH that is specifically focused on cancer, where we have actually enacted essentially a precision medicine application in dogs to show that it was doable. We could collect samples, send them off for sequencing, make recommendations, and send it back for those patients in a clinically relevant time frame. And I think expanding on that is really important to develop a framework for those kinds of translations, because it's a real challenge. The other real challenge is the way that drugs are monetized at this point, because if you have a large population of cancer patients and there is evidence early on that only 10% of those patients will benefit from that drug, it's very difficult to move that drug forward in the commercial development setting, because unless it's a blockbuster, they're not very interested in that. And so we're going to have to figure out ways that we can monetize non-blockbuster drugs, and that's not something we can probably do ourselves. That's something that's going to need broader societal funding.
Okay, we have one more question. This will be the last one then.
I'm Celestin Yuan from UMKC, the second unofficial rep from the Kansas City. So my area of interest, I'm in the School of Pharmacy, first of all. So I teach drug delivery systems. And the way I see precision medicine going on, collaboration with drug delivery, is to have maybe precision delivery systems. So I'm here to maybe be part of the discussions. Anyone has a new molecules. One of the goals, for example, for our group is to, in the future, can we take the genetic profile of the patients and before they go out from the hospital, have a delivery system uniquely tailored for them. So that's something that we are interested in this discussion.
Agree. That's true. We need that. Anyone want to comment?
At the working lunch. Lots of opportunities at the working lunch to get everybody together, so make sure you're there.
So let's thank our speakers one more time.